Meiosis is carried out in two rounds called Meiosis I and Meiosis II. Why is that so? That's correct. To obtain 4 haploid cells with half the number of chromosomes compared to the parent cell. Let's begin with Meiosis I first. Meiosis I is divided into 4 stages. They are prophase I, metaphase I, anaphase I, and telophase I. We will list down the changes observed in the cell during these stages step by step. Let's begin with prophase I. It's the longest phase of Meiosis I. The phase is further divided into 5 substages. They are leptotene, zygotene, pacotene, diplotene, and diakinesis. Leptotene is the first substage. In this phase, the cell begins condensation of the chromatin, making it short and compact. The next stage after leptotene is the zygotene substage. Here, the homologous chromosomes form a pair called the synapsis. The new structure that is formed after the union of 2 homologous chromosomes is called a tetrad or a bivalent. After forming bivalents, the cell enters the next substage called the pacotene. Here, the bivalents exchange gene segments between the non-sister chromatids of homologous chromosomes. And can you tell me what this process is called? That's the right answer. It's called crossing over. And the location where the exchange takes place is called chiasma. After successfully interchanging the genes with one another, the cell enters the next substage called the diplotene. In diplotene, the bivalents unbutton themselves, resulting in the separation of the 2 homologous chromosomes. But here, the tricky part is that the homologous chromosomes are still anchored at the chiasma. Finally, in the last substage called the diakinesis, the homologs get completely unhooked and regain their individual identity. The nuclear membrane and the nucleolus completely disappear by the end of prophase I. Now let's move ahead with the next stage of Meiosis I, that is metaphase I. The homolog pairs align on the equator of the cell with random orientation. This arrangement is called the metaphase plate and is a result of independent assortment. In the successive phase, that is the anaphase I, the homolog pairs separate from each other and move towards the opposite poles. Finally, the cell undergoes the last phase, that is the telophase I, along with the first cytokinesis. The 2 new cells obtained are now haploid in nature. The telophase I, followed by cytokinesis, helps in the formation of a nuclear membrane surrounding the chromosomes and a nucleolus as well. The daughter cells are haploid, which are